Every week, or every other week, when you turn on this program, you're in search of something new. What am I going to learn today? For most of you out there, you may prefer to listen to shows that align with your interests. If you like neurocritical care, you probably really liked Dr. Maciel's show on the ictal-interictal continuum, episode 145. Or if you're into neuromuscular disease, you probably liked Dr. Levinson's show on myotonia, episode 134. Or maybe you prefer listening to something you had no idea about. You stepped outside of your comfort zone and maybe checked out Dr. Ross talk about eye pain, episode 121. Stuff that you probably never thought you would want to read or learn about. But either you had to know it, or you thought it would be more interesting to hear in an audio form. Well, this week on the program, I'm definitely stepping out of my own comfort zone. I'm almost embarrassed to say this, but I don't think I've been that inspired by the clinical aspects of cognitive neurology, or cognitive pathophysiology, or outpatient medicine. So in light of us all coming together and challenging ourselves to learn something new, I thought I'd do a show on today's topic. And you know what? It's actually pretty amazing. It takes me back to my college years, when I was first exposed to the fascinating world of neuroscience and the cellular and molecular mechanisms that contribute to neuropathology. So this week on the program, the role of Astroglia in Alzheimer's disease, from our current understanding of their biologic roles to the aberrant circuitry that underlies neurodegenerative disease, and eventually the implications for future therapeutics. Stay with us. Even if you're not a cognitive neuroscientist, you know that Alzheimer's disease is a fatal neurodegenerative condition that's characterized by the buildup of abnormal proteins throughout many brain regions. So let's just imagine this cellular milieu. When we're talking about the neurodegeneration of Alzheimer's disease, we're talking about widespread loss of pyramidal neurons in the frontal, the temporal, and parietal lobes, as well as neuronal loss of the entorhinal cortex of the hippocampus, the thalamus, amygdala, and regions of the brainstem, like the nucleus basalis of Meinert, which produces acetylcholine and is the therapeutic target of cholinesterase inhibitors like rivastigmine and donepezil, and the noradrenergic nucleus, the locus ceruleus. Basically, Alzheimer's is everywhere. All of these neurons slowly and unstoppably die due to toxic production of intracytoplasmic neurofibrillary tangles made up of the hyperphosphorylated microtubule protein tau. Let's see if I can find a sound effect for that. Yeah, that works. While our beta amyloid plaques over here aggregate outside of neurons. Pathologic studies have shown that the burden of the intracellular neurofibrillary tangles correlate best with the severity of the dementia, and we can quantitate this with CSF testing and advanced neuroimaging. Phosphorylated tau and total tau levels increase in the CSF dramatically in patients with Alzheimer's disease, and these elevations can vastly predate the onset of clinical symptoms. So there's enormous potential for this biomarker in clinical trials. Amyloid PET imaging has also been shown to have a similar sensitivity and specificity for detecting preclinical AD, at least according to one Swedish cohort that was published in Neurology in 2015. And you can find all these references in this week's show notes. So we have these protein aggregates, right? These pathologic proteins. They're building up either inside the affected neurons themselves, in the case of neurofibrillary tangles, or outside of the neurons, as is the case for amyloid beta plaques. What does any of this have to do with glial pathology? Well, we can't really answer that question yet, because first we need to establish the roles of astrocytes and supportive tissue in central nervous system function. Then we can dive into more of the pathophysiology and the future therapeutic targets. As you know, astrocytes are among the most important and mechanistically diverse glial support cells within the central nervous system. Astrocytes are like the Renaissance men of the brain. They're cells with many tasks and many talents, and when they go missing, social norms begin to disintegrate. Astrocytes are responsible for GABA and glutamate reuptake from the synaptic cleft, which is crucial for the prevention of neuronal excitotoxicity. These cells assist with synapse formation and modulate neural connections, helping to eliminate unwanted synapses. Astrocytes are integral components of the blood-brain barrier, and they regulate excretion of metabolic waste through the glymphatic system. They keep the brain an immunoprivileged site, relatively speaking. And if you've ever seen an astrocyte under a microscope, you can see that it's got this star-shaped appearance, with the soma at the center, and these projections that extend in multiple directions, deploying in feet onto the small arterioles of the brain, which ultimately form the blood-brain barrier. It's pretty elegant, actually. In the setting of neuronal activity and release of glutamate, the astrocytes not only take up the excess glutamate via endocytosis, but some of this glutamate also activates astrocyte metabotropic glutamate receptors. And it is the job of these receptors to signal the production of arachidonic acid metabolites, and through a cascade of events, this will trigger the influx of calcium into the distal astrocytic infeet, with the release of various vasoactive substances that ultimately promote local arteriolar vasodilation and increase delivery of oxygen and nutrients to these metabolically active brain regions. Scientists call this the neurovascular unit, and this succession of events, which leads to increased regional blood flow, takes place over 1 to 2 seconds, which is really amazing. In addition to each of these functions, astrocytes also facilitate delivery of nutrition from these arterioles to adjacent neurons in the form of lactate, and these neurons then convert the lactate into pyruvate for energy metabolism. If you think about the sheer number of supporting cells in the central nervous system, in the supratentorial brain, glial cells outnumber cortical neurons 4 to 1. And in the brainstem, where, as I mentioned earlier, there's additional Alzheimer's pathology, glial cells outnumber neurons 11 to 1. Of all the glial cells, our buddies, these Renaissance astrocytes, they account for 20 to 40% of glia and are the most abundant cell type in most parts of the brain. I bet you might not have known that. Then there are also the microglia, which comprise about 10 to 20% of these glial cells in the adult brain, and they serve as the immune cells of the nervous system. In the presence of cell death, microglia migrate toward diseased brain regions and trigger an immune response in an attempt to clean up the injured tissue. Then you've also got your oligodendroglia, which establish and maintain myelin sheaths. You've got your ependymal cells, your radial glia, and so on and so forth. But we're not going to talk about these cells today. Returning to the astrocytes, however, they play a crucial role in the pathophysiology that underlies Alzheimer's disease. Even among the earliest histopathologic descriptions of AD, reported by Alois Alzheimer in 1910, the presence of glial cells was a requirement for senile plaque formation. In animal models of Alzheimer's disease, reactive and atrophic astrocytes are found long before plaque and tangle formation occur. Gene studies have also shown that most of the total risk for developing Alzheimer's disease is due to genes that are solely expressed in glial cells. Apolipoprotein E you'll certainly recognize, but then there's also clusterin, or ApoJ, fermentin, ATP-binding proteins involved in lipid metabolism, ATP-binding cassette subfamily A member 7, apolipoprotein E, sortilin-related receptor 1, proteins involved in the central nervous system's immune response, triggering receptor expressed on myeloid cells, also known as the TREM2 receptor, major histocompatibility complex, IL-1 receptor-associated protein, and a number of other functions, protein tyrosine kinase 2 beta, kinase anchoring protein 9. When it comes to Alzheimer's disease, a number of these things go haywire. With beta amyloid plaque formation, as the hypothesis goes, the amyloid precursor protein is cleaved by alpha and beta secretases. A byproduct of the beta secretase, or the BACE1, BACE1 pathway, is the amyloid beta peptide monomer, which will spontaneously aggregate into insoluble oligomers. In the healthy human brain, beta secretase is expressed exclusively by cortical neurons. However, under chronic stress, or with excess activation of astrocytes, as we see in Alzheimer's pathology, the astrocytes begin to express beta secretase too. So not only are they going to start reacting to neuronally produced beta amyloid plaques, but they themselves will produce the plaque as well. But before all this even happens, at the very beginning of astrocyte activation, we've got our quiescent astrocytes, the Renaissance glial cells. And they've begun to detect increasing levels of amyloid beta 42 oligomers, and now they're trying to engulf these plaques. In addition to endocytosing these amyloid plaques, the reactive astrocytes are now responding to all the associated damage that the amyloid plaque has triggered. You've got reactive oxygen species, tau hyperphosphorylation, and the ensuing inflammatory deterioration of neuronal synapses. The exact sequences to how this happens is not perfectly elucidated, but animal and cellular models have shown that the quiescent, or these non-activated astrocytes, are somewhat instigated by local microglial inflammatory signaling. The nearby microglia secrete proteins like TNF-alpha, IL-1-alpha, and C1q, thereby activating astrocytes and further promoting inflammatory glial machinery, including an appropriately named protein named RAGE, which I'll talk about later, all of which are critical components to the neurodegenerative process. Now exposed to these pro-inflammatory factors, the reactive astrocytes will begin to ramp up production of NF-kappa B and complement component 3, which are toxic to cortical neurons, and this leads to local pyramidal cell death. The astrocytes then begin to release their own cytokines, interleukins, nitric oxide, and other pro-inflammatory molecules that exacerbate the evolving local immune response. While doing this, our reactive astrocytes will fail to maintain the integrity of synaptic connections, probably for good cause, since these cells are detecting local neuronal cell death. They've begun to phagocytose the freely wandering neurotransmitters that have escaped synaptic clefts, which is now building up to excess levels in the extracellular environment, and this leads to adjacent neuronal cell activation and excitotoxicity. As a secondary, and a likely protective response in order to contain that region of cell death, the astrocytes begin to exocytose the glutamate that they had taken up from local synapses, which further perpetuates this excitotoxic response, leading to additional neuronal death. Now this all sounds kind of bad. I mean, how would such a pathologic remodeling strategy persist throughout human evolution? Well, there are benefits to this type of reactive gliosis. With inflammation and scar formation, astrocytes and the associated glial tissue are attempting to isolate these pathologic neural glial units. They're trying to remodel the blood-brain barrier and promote the clearance of diseased tissue. When there's impairment in this astrocytic response, we're left with conditions like Wernicke's encephalopathy and amyotrophic lateral sclerosis, where glutamate excess is not adequately managed. In addition to all these pathologic responses that we see in Alzheimer's disease, astrocytes are critical to neurodegeneration as they are the main cells within the central nervous system that express apolipoprotein E. At the cellular level, ApoE is responsible for cholesterol uptake. One variant of ApoE, the ApoE4 variant, is the most common genetic risk factor for the development of late-onset Alzheimer's disease, with as many as 40 to 60% of AD patients carrying one or more ApoE4 alleles. Now, I'll say that it's not a guarantee that you'll develop AD if you express the ApoE4 variant, but having 2 copies of this allele makes a person 20 times more likely to develop AD. But how does this translate at the cellular level? So we have these ApoE4 expressing astrocytes, and unlike ApoE3 astrocytes, for example, those expressing ApoE4 are much worse at phagocytosing and degrading the surrounding extracellular amyloid beta plaques. ApoE4 expressing astrocytes may also be responsible for more seeding of amyloid beta plaques, and through a number of other mechanisms may lead to breakdown of the blood-brain barrier, which is known to activate the NF-kappa B pathway and facilitate complement release and other resultant CNS inflammatory responses, all of which can lead to neurodegeneration. And just to bring it back full circle, these inflammatory mechanisms further activate quiescent astrocytes, leading to impaired cortical signaling, excitotoxic buildup of local neurotransmitters, and impaired amyloid plaque uptake, and so on and so forth, which is how this cyclic neurodegenerative process self-perpetuates. So for those of you out there like me who aren't so histopathologically inclined, what does all this mean? What are the clinical implications of understanding the relationship between astroglia and Alzheimer's cellular pathology? Does knowing any of this allow for some sort of therapeutic intervention? Let's start from the top, at the production level of amyloid plaques. Here, beta secretase is an early enzyme in the metabolic machinery that leads to amyloid beta 42 plaque formation. As you'll recall, beta secretase, or BASE1, is one of the enzymes responsible for cleaving amyloid precursor protein and generating our amyloid beta 42 monomers. Inhibitors of BASE1 have been studied in various preclinical and clinical trials, drugs like verabecestat, solanezumab, lanabecestat, and some compounds that are only known by a string of numbers and letters. For the most part, each of these inhibitors have been shown to penetrate the central nervous system, and they can effectively and dose-dependently reduce the CSF concentrations of beta amyloid. However, the clinical outcomes have not seen as much success.
Researchers found that the new promising Alzheimer's drug, Verbeek, did not work. Dr. Roger Perlmutter...
One recently published Phase 3 randomized controlled trial of Verbeek from 2018 was terminated early due to futility, and also due to the high rate of adverse events with the treatment arm: rash, falls, suicidal ideations.
This is a huge disappointment for Alzheimer's researchers. They were really hoping...
These results were published in the New England Journal of Medicine. Very unfortunate. An unrelated BACE1 inhibitor, known as LY2886721, also showed some promise in preclinical models. However, the Phase 2 trial was terminated early due to the unexpected finding of abnormal liver function testing. A more direct approach to amyloid beta reduction has been the development of monoclonal antibodies that are directed against the amyloid plaque.
And now, news about a drug that may slow the progression of the disease. Dr. Ronald...
You've probably heard about this.
An antibody that showed reduction of amyloid accumulated in the brain during clinical drug trials. It was
the Alzheimer's disease vaccine. These would be agents like aducanumab, cantinumab, crenizumab, and some other names that I don't expect you to remember after listening to the program today. Thus far, these immunotherapies have been shown to reduce the burden of amyloid plaque in Phase 1 and Phase 2 trials, but as with the BACE1 inhibitors, trials using solanezumab and bapineuzumab failed to demonstrate a clinical benefit. In spite of these failed studies, there are 3 other monoclonal antibodies that are currently in Phase 3 trials, which showed promise in their Phase 1 and Phase 2 studies, and these trials are expected to reach completion in the next 3 years. And if there's anything that we should take away from other neuro literature, like stroke literature, thinking about things like tissue plasminogen activator or mechanical thrombectomy or PFO closure, we shouldn't just give up on these potential interventions because related therapeutics have failed to demonstrate clinical efficacy. While they are all antibodies that are directed towards the same target, each of them are unique agents, and all it takes is for one of them to work. Regarding inflammatory targets, the Alzheimer's disease literature is yet again plagued by a number of negative randomized controlled trials. One early trial of indomethacin suggested a benefit for cognitive decline, but these data have not been replicated on subsequent attempts. Other NSAIDs, and statins, and hydroxychloroquine, and corticosteroids also failed in human trials of AD. One trial, using naproxen for the prevention of AD, has shown some promise. The ADAPT investigators, after originally showing naproxen to be detrimental to patients who had symptomatic AD, they actually showed a significant reduction in the incidence rate of AD among asymptomatic persons. The study was limited, however, in that it only randomized 2,000 individuals, and it needs replicating. But the results are curious, and they are potentially promising. More specific targeting of the CNS inflammatory response, for instance, with an inhibitor of calcineurin or the nuclear factor of activated T cells, has been shown in cellular models to reduce the astrocytic inflammatory response, and they reduce beta amyloid plaque formation, but we will need human trial data to validate any clinical effect. Within the astrocytes themselves, after engulfing the amyloid beta oligomers, there is a downstream cascade of intracellular responses. Targeting these pathways has shown promise in cellular and animal models. Consider the phosphodiesterase inhibitors, for example. Drugs like roloprem and sildenafil have reversed cognitive and memory deficits in animal models of Alzheimer's disease, but we still have no human trial data to support their routine use in AD just yet. In addition to PDE inhibition, attempts at preventing reactive oxygen species formation, which is a natural consequence of excess amyloid beta 42 plaque formation, this is also a potential target in AD. Antioxidants like vitamin E, vitamin C, and carotenoids have been studied, and like I mentioned earlier in episode 135, Brain Food, they haven't been definitively shown to prevent cognitive impairment or slow the progression of Alzheimer's disease. Statins, on the other hand, by reducing the synthesis of cholesterol, have been associated with improved cognitive performance and memory recall. The thought is that, through mechanisms beyond just cholesterol formation, they attenuate glutamate neurotoxicity in cortical neurons, and they suppress the inflammatory response of microglia and our astrocytes, but only at lower doses. In addition to these generally anti-inflammatory and antioxidant pathways, the receptor for advanced glycation end products, awesomely referred to as RAGE, I mentioned this one earlier, this receptor is also a potential target. To simply put it, once the insoluble beta amyloid oligomers and neurofibrillary tangles form, They sit around for a long time, and they're subject to incidental glycation, which results in plaque and tangle cross-linking, and this leads to more neurotoxicity and progression of cognitive impairment in AD patients. Astrocytes and other glial cells express the RAGE protein, the receptor that binds to these glycation end products, and ultimately, the interaction between RAGE and the neurofibrillary tangles and amyloid plaques triggers the additional hyperphosphorylation of tau. This is the mechanism by which RAGE overexpression can facilitate AD progression. With that said, preclinical data have shown that the small molecule RAGE antagonist azaliragon can reduce beta amyloid plaque formation and deposition. Furthermore, in a phase 2b trial of patients with mild AD, after 18 months of treatment, azaliragon was associated with improved cognitive performance and slower cognitive decline when compared to placebo. But unfortunately, we still haven't gotten this one right either. The phase 3 trial, Steadfast, recently terminated early due to futility. It may have been on the basis of too low of a dose, though. These results are so far unpublished, so we'll just have to wait. There are many other therapeutic targets that I didn't have time to cover today, but hopefully this gives you a sense for the interconnection between pathology and pharmacology in this neurodegenerative condition. At this time, the summer of 2019, the only available drugs for AD, memantine and cholinesterase inhibitors, they target symptoms of disease, not the proximate cause. We have no disease-modifying therapies to prevent this neurodegenerative cascade of events, and nothing to slow it down once it's started. Alzheimer's disease accounts for more than 80% of all dementia cases worldwide. There are more than 5 million people who are living with AD. And by 2050, according to data from the Alzheimer's Association, a new case of Alzheimer's disease will be diagnosed every 33 seconds. Then again, by 2050, these Alzheimer's patients won't have a place to live, because apparently Earth won't be here anymore. Scientists have now estimated that we will have destroyed our planet through excess consumption of limited resources, the effects of climate change, depletion of potable groundwater and aqueducts, and 1,000 other things. But in the meantime, neuroscientists are hard at work trying to design better, safer, and more targeted treatments for Alzheimer's disease. And with any luck, we'll have solutions for both climate change and dementia in our lifetime. You know, I really learned a lot by preparing this episode. I don't see a whole lot of AD in my own practice, and I was quite surprised at all the various therapeutics in the preclinical and clinical pipeline. Even if you don't remember all their names and their mechanisms of action, at least you can take away that we have many types of unique small molecules, monoclonal antibodies, and therapeutic targets, which may one day reduce the progression of AD, or even eliminate it entirely. Only time will tell. And if you didn't get enough cognitive neuroscience out of today's program, we've also got shows on language localization. See the show that was titled Aphasiology, Neural Networking in episode 63, and episode 113 on the management strategies for hypoactive delirium. The BrainWaves Podcast is produced by myself, Jim Siegler. Music for this week's program was courtesy of Andy Cohen, Brendan Kinsella, Q-Cute, Damiano Baldoni, Kai Engel, Nocturnum, all under Creative Commons license. Sound effects were by Mike Koenig, BlastFX.com, Carolyn Ford, Robert V., and Daniel Simeon. In the next couple of weeks, we got shows on the Tourette syndrome, neurosarcoidosis, and the link between infectious and autoimmune encephalitis. Keep an eye out for them. I'm Jim Siegler for BrainWaves. Thanks for listening.